Hello everyone, welcome back to AS Biology with Dr. Demi. As usual, I am Dr. Demi, and in this video I will take you through antibodies and vaccination. This is the penultimate video for the AS Biology syllabus, which means that after this video we only have one more left to go, and we will call it a wrap, except you have questions or you need some sort of an explainer video and I'm able to do that, then definitely I will try as much as possible to put those out for you. But for now, we are very close to the end, and I hope that you guys are excited. I hope you've been revising well for your exams and you are looking forward to them. If you have any past questions you're working on at the moment and they look a little bit blurry or you just don't understand what's going on, by all means post them in the comments and I promise to get back to you with some sort of explanation. But before then, try to watch this video on antibodies and vaccination, and I hope that you enjoy it. So when people hear antibodies, or rather should I say when my students hear antibodies, I asked them before I showed them the slide, what comes to mind? And some of them said, okay, things that look like enzymes with active sites and all of that. And that's not entirely wrong, but I don't think they were expecting to see something that has this shape over here on the right. So antibodies are Y-shaped molecules. You can see that over there. And they are actually globular proteins. And I think this got students a little bit confused because if it's globular, why does it look like that? But antibodies are globular proteins. They have a quaternary structure. Remember from chapter 2, we said a quaternary structure simply means that the polypeptide chains are 2 or more. So if a protein has 2 or more polypeptide chains, it is said to have a quaternary structure. In the case of antibodies, antibodies have 4 different polypeptide chains. So they have 2 light chains, which are these small chains over here. I'm just going to circle that with a red pen. So these are the light chains, and they also have the heavy chains. So you can see over here, the blue sections are the heavy chains. I just noticed that I made an error trying to circle the light chains. The light chains are actually just the green sections over there. You have the binding site as well. So the binding site is where they would bind to something like an antigen, and they have something called a hinge. So they form a group of plasma proteins that are called immunoglobulins. And again, remember 4 polypeptide chains. It is important for you to know that the polypeptide chains are held together by disulfide bonds. Now, this takes you back again to chapter 2, and I often tell students that whatever you learned previously in AS biology and even as you go on to A level becomes more and more important because you will find it staring at you in the face when you get into new topics. So don't discard anything you've learned previously. They also have the binding sites are also called variable regions because they are different based on the antibody. Remember we said that B cells, like each type of B cell would make a specific type of antibody, and so that means that the antibodies differ based on the antigen that B cell is allocated to. The hinge region, which is this region over here, think of it as the hinge of a door, right? So when you have a door in your house, you open it, it turns on its hinges. You don't move the door, you don't carry the whole door out of the way when you're trying to open the door and then fit it back. You just open it and it turns on its hinge. So the hinge regions help to make the antibodies flexible. It's important that you know the structure of an antibody because these are the kinds of questions that come up in your exams where you might be asked to label the antibodies. Now, what happens when you get an infection? How does that work with the antibodies? There are 2 kinds of responses that your body is likely to elicit, and that's either a primary response or a secondary response. A primary response simply means that your body is encountering an antigen or a pathogen for the first time. So we call that first time initial exposure. So over here, it means that when that antigen hits the body, the concentration of antibodies would be low because the antibodies or the B cells have never seen this antigen before. That means that they then have to go through the process of learning about the antigen, making copies of antibodies that can address the antigen, and what that then means is that the antibody concentration in a primary response is quite low. Yeah, that means that it's really low. But what happens if after you get the antibody response, you become a little bit better, and a few months or maybe a couple of years down the line, you're exposed to the exact same antigen again? In this case, you would have what we call a secondary immune response. The secondary immune response means that the body knows the antigen. The memory B cells, remember we discussed them in the previous video, have learned about this antigen. So the moment they see it, they're able to send the signals out immediately to say, we are dealing with this antigen that visited us about 6 months ago. It's back again. We need to deal with it. And what the body will then do is that it would simply reproduce the plasma cells or the antibody cells that had been made before, so that the cells are able to go and attack the antigen. As a result of that, the secondary immune response is faster because the immune system is not starting from scratch. It's starting based on the knowledge it already has of the antigen. Now, there are two types of immunity when we speak of immunity. You can have active immunity or you can have passive immunity. Active immunity is simply immunity that is developed in response to the pathogen or a bacteria. The pathogen can be a bacteria, virus, whatever it is. So you can have natural active immunity, which means that you get infected. So say, for example, you haven't seen your cousin in a long while. You've eventually met up with your cousin. You give your cousin a hug. Your cousin has the flu and somehow, she breathes the droplets onto your skin. And now these droplets sort of get into your airways and now you'd come down with the flu. Your body learns how to fight that, and in the process it builds immunity. We call that a natural active immunity. Active because it is in response to an infection, and natural because you simply got infected. You can also have artificial active immunity. Artificial active immunity would be immunity you develop as a result of vaccination. So interestingly, I have students who get vaccinated for the flu every year. And I remember a particular student came to me and said when she got vaccinated with the flu vaccine, she fell ill for a few days and she felt like, oh my goodness, this vaccine did not work. It simply made me ill. But what the vaccine was simply doing at that point in time was that it was trying to get her body to respond to what was the virus or what the virus looked like. So she was developing artificial active immunity. You can also have passive immunity. An example of passive immunity that is natural is the immunity that a mother passes on to her baby. So it is very important. That's why it's very important for a woman who's carrying a baby to be in very good health, because she passes her immune cells into the baby and helps the baby develop immunity. It's also important when she's breastfeeding because through breast milk, she's able to pass on all of these immunity cells or maternal antibodies as they're called to the baby. You can also have artificial passive immunity, and that is when you have artificially manufactured antibodies injected into the body. And I think in our next video we will discuss how these antibodies are made. They are called monoclonal antibodies. So we'll learn a bit about that in the next video, which is the last video, by the way. So what do antibodies do? And usually students get shocked here because I think many students expect that antibodies go there and they secrete some sort of destructive substance into pathogens and the pathogens die and they do all of these fun things. But that's not usually the case. Antibodies combine with like viruses or even bacterial toxins and prevent them from entering any cells or from damaging them. So it's like they become a weight on the pathogen. They attach themselves to it, and as a result of that, the pathogen is unable to act as it normally would. It's unable to attach attach where it normally would. Sometimes antibodies will attach to the flagella of bacteria. Remember what flagella is. Flagella is like that tail-like structure that bacteria use when they're trying to swim through a medium. So remember from the structures of prokaryotes, again, go back to chapter 1 and have a look at that. So they attach to the flagella, and that makes the flagella, it makes it slower for the bacteria to move, makes the bacteria less active, and makes it easier for phagocytes to then come and engulf the bacteria. Antibodies can also cause clumping together of bacteria, so they can collect the bacteria together and clump them into bigger clusters so that they are unable to spread around the body. And as a result of that, again, the phagocytes are easily able to find the bacteria and just, chop them up, swallow them, digest them, and that would be the end of the story. Some antibodies can make holes in the cell walls of bacteria. So this, I think, is what many students tend towards, because they're like, yes, yeah, that's what we're talking about. So some of them can make holes in the cell walls of bacteria. Some of them are able to coat the bacteria so that the phagocytes can easily recognize them. So sometimes bacteria come into your body and they try as much as possible. They're not explicitly or distinct. They're not distinct. I think that's a better way to put it. It's hard for the body to say, oh, there's a bacteria, but the antibodies are able to then spray them. It's like, imagine just painting someone pink and telling the person, okay, well, go walk in the mall. Obviously, everybody will see that this person is pink. In the same way, antibodies can coat bacteria so that phagocytes are able to recognize them and ingest them, so digest them through endocytosis. And sometimes they combine with toxins that are released by pathogens, and they make the toxins harmless. And these kinds of antibodies are the ones we call the antitoxins. So that said, that's more or less a revision of antibodies. Again, remember, this is just a revision channel. So by all means, make sure you go and look in your textbooks, in your school notes, so that you're sure that you've gained all the information that you need to know for your exams. So I will now go on into vaccines. And again, this is exciting. I'm recording this just after the vaccine for the COVID-19 pandemic has been released, the Pfizer vaccine. So it's very interesting that I'll be discussing this now. But what a vaccine is a preparation that contains antigens that would then be used to stimulate an artificial immune response. So let's say, for example, I'm going to use a red pen. So let's say, for example, here is a virus. Okay, this virus has spikes just like the COVID-19 virus. And so when the body sees it, let's say the body has never seen it before. So now we need to vaccinate people with this virus. Let's assume COVID-19 was not so deadly. Let's say it was just a very mild virus, but all the same, we want to vaccinate people. We can actually take this live organism and introduce it into the body. What that would do is that it would cause active immunity, because what we've done is we've more or less infected the person. And that then means that the cells, the natural immune system, will start to work towards addressing this virus because it's never seen it before. So it's like trying to introduce a copy into the body to say, here's what this infection would look like. And then the body is prepared. So when the actual infection comes, the body is able to fight it, because again, they're the memory B cells, they're the other B cells, the plasma cells that secrete lots of antibodies. You also have the killer T cells, you have the helper T cells. All of them have learned how to recognize this particular image. So what this means is that a vaccine can be a live microorganism. It can also be a dead microorganism, which means that in this case, there is nothing inside of the spike, only just this shell is made. There is nothing on the inside, there is no DNA, there is no RNA. And so just by introducing the external region of the virus or the pathogen into the person, the body is still able to recognize what it looks like on the outside, because it doesn't really matter what it looks like on the inside if the outside already results in a very good immune response. You can also have a harmless version, which is an attenuated version, and then you can have a harmless form of a toxin, or you can have a preparation of surface antigens. Just for interest's sake, it is here that I think I should tell you that the Pfizer vaccine has been said to be an attenuated version. of the virus, in the sense that it only has the spikes on the outside. There is nothing inside, which means that once this is introduced into the body, it will elicit an immune response, and the body is then able to say, oh, okay, this is what this virus looks like. Obviously, it destroys that spike, just that surface antigen. So if the COVID-19 virus were to then penetrate the body is again able to recognize it very quickly before it can cause any harm, and is able to destroy it as quickly as possible. Some vaccines are highly effective, so sometimes you get a lifetime injection and that's it for life. Others are not that effective, so you need what we call a booster injection, which means after a certain period of time you need to go get another injection so that you can get a secondary response for greater protection. Now, vaccines are not without their problems. And of course, again, if you're living the time of the pandemic, you're aware that many people are very, very skeptical about going to take a vaccine that they say has been developed within a very short period of time, which is true. But also the history of vaccines or problems with vaccines as well has sort of made people more skeptical of this particular one by Pfizer. And I think there's another one by Moderna, and they use the same technique. So in some cases, there have been poor responses to vaccines. There has been, like a report I read was talking about how people had poor responses to the polio vaccine and ended up developing polio instead of being vaccinated against it. Not all vaccines obviously cause this response. It can sometimes be due to a defective immune system that doesn't produce normal T and B cells, which means that if you were to use a live version of the vaccine on this kinds of people, what would happen is that they would not produce normal T and B cells, so there would be no memory cells, and that means that the virus will then be able to ravage the body and cause the disease that you're trying to vaccinate against. Sometimes also, if there's malnutrition where people don't eat enough proteins, that can result in a poor response to a vaccine. There can also be a live virus and herd immunity issue. When some people are vaccinated with a live virus, they might pass that virus out through their feces or even like through their urine. Some people just pass out viruses even through their body fluids. And as a result of that, other people might get infected. And so this is important because it's the reason why large groups are vaccinated at once so that everyone is immune at the same time. This is what we call herd immunity, because it is immunity that prevents transmission within the community. Even if a person were to pass out the virus, the live virus in their feces, it doesn't result in any other person getting infected. You can also have antigenic variation. So some viruses mutate very frequently. For example, the HIV virus. When people say, how come we don't have a cure for HIV or a vaccine for HIV? That is the response. The fact that HIV mutates very quickly, which means that by the time a new virus, a new vaccine has been developed or some kind of cure, the virus has already changed its structure. But the good news is that I was able to meet with some amazing researchers. You should check out Prof. Karim. He's an HIV researcher in South Africa, working with Dr. Bruce Walker, Dr. Howard Heller. These are men who are working on the HIV virus and also just developing preventive measures. So you might find that some of them they've developed a vaccine that would be going into trial very soon. So minor changes in a viral antigen can stimulate a secondary response. And that's usually called an antigenic drift. So if you have a major change in a virus, that means in its DNA structure or whatever it is, then that is an antigenic shift, and in that case you might need to find a whole new vaccine altogether. You can also have antigenic concealment as a problem with vaccines. Some pathogens hide inside cells, so that means that their antigens are not easy to see, which again makes it important that the killer T cells are able to go and patrol the body and scan the cells of the body to see if they are hiding or harboring any pathogens and their antigens, and to destroy such cells. This is the last part of this video. I'm just speaking about smallpox, which was caused by the variola virus, and smallpox has been eradicated successfully. And this image here from the World Health Organization shows the years during which it was eradicated. You can see there in South America, it was in 1971. In Africa, it was in 1977. And the good thing about this virus was that it did not mutate. And so because it did not mutate, it was easy to develop a vaccine in response to it. The vaccine was made from a similar live virus, which meant that when the smallpox virus came into the body, because the body had seen a very similar virus, the body was able to fight against it. The vaccine was freeze-dried, so it meant that it could survive very high temperatures for up to 6 months. So in regions that are really, really hot, for example, this vaccine just simply worked. There was also a herd immunity approach to the vaccination, in the sense that communities were vaccinated at the same time. So that meant that it was hard for anyone to get sick, even if they passed out the virus in their feces or in water or through any of their droplets. The vaccine was also very easy to administer, and it did not infect animals, because sometimes when a vaccine crosses over and starts to infect animals, that itself creates a different set of problems for the people who are conducting the vaccinations, because it might mutate and become some other kind of virus that they can't recognize. So that's it on antibodies and vaccinations. If you have any questions, as usual, please post them in the comments, and I will try to get back to you as soon as possible. The last video, which would be the last part of the AS biology syllabus, will speak more about monoclonal antibodies, and I'm sure you're excited about that. It will also speak of autoimmune diseases. So yeah, make sure you watch that and have lots of fun. Goodbye.